Kevin, Logan, and all the MCI team for giving us the opportunity to share with you our male contraceptive research in South America, more specifically in Argentina. And as you know, 15% of couples in reproductive age have fertility problems, indicating the need to develop new methods for both diagnosis and treatment of infertility. But at the same time, there is a 40% of unwanted pregnancy, 20% of which ends in abortion. And these numbers increase a lot significantly for developing countries. So indicating the need of also developing new contraceptive methods that meet the different needs and preferences of people in different parts of the world and at different stages of their reproductive lives. Now, which are the current contraceptive options? As you know, for women, we have several hormonal methods, the pill, the IUD, diaphragm, cervical caps, etc. But for men, we only have the condom and vasectomy, which cannot be considered really a contraceptive method because you have only 50% reversion and you never know in which percentage it will fall. Now, considering there is 60 million vasectomized men in the world, it is clear there is a niche in this field. The reason why development of methods for male fertility regulation have been an important topic since the 70s in different international fora like WHO, NIH, Rockefeller Foundation, Conrad, and more recently, the Bill and Melinda Gates Foundation and MCI, with the idea to emphasize the responsibility and promote the participation of men in family planning, meaning whether or not to have children, when, how many, and how spaced. Now, although this will involve important cultural changes, the good news is that the level of acceptability is at least 50% among men and higher among women. Now, if we want men to use contraceptive methods, we need to give them more options. So why to develop male contraceptive methods? First, because family planning must be shared. It is not a woman issue, it is a couple issue. Second, because of health reasons. As you know, women use contraceptives for 35 years, from menarche to menopause. Third, because it's a male right. We have to stop thinking that male contraception is just to help women and to begin thinking that it empowers men and the couple. And finally, because there is a niche according to the high percentage of vasectomized men in the world. Now, the development of male methods involve, of course, hormonal and non-hormonal approaches, which have the advantage to avoid the many side effects inherent to the use of hormones. And this is not a minor point because side effects are much less tolerated by men than by women. So the non-hormonal methods, which are at the more basic research level, involves interference at three different levels, mainly these three that I will show you. Production in the testes, that is spermatogenesis, interference with the release of a sperm through the vas deferens during ejaculation, or interference with the maturation process that occurs in the epididymis, this organ shown here in yellow. And this is based on the fact that sperm that leave the testes are immotile and unable to fertilize the egg, requiring to undergo a maturation process that occurs while sperm are passing through the epididymis to acquire the fertilizing ability. And this occurs mainly as a consequence of the association of epididymal proteins to the sperm plasma membrane. So the epididymis is considered an excellent contraceptive target because there will be no interference with the sperm or hormone production, important, critical for libido, but a specific interference with the acquisition of a sperm fertilizing ability. And the idea of having an ejaculate without the sperm fertilizing ability is represents a safe, attractive, and promising approach. The reason why epididymal proteins become very good targets for fertility regulation. Based on this, the aims of our lab have been to study the role of epididymal proteins in sperm maturation and fertilization, and also to explore their potential use for fertility regulation. And we have been doing this using a group of proteins called CRIS for Cysteine Rich Secretory Proteins that contain 16 conserved cysteines, 10 of which are located in the C-terminal region of the molecule called CRD for Cysteine Rich domain, and which has ion channel regulatory activity. And the remaining 16 are in the N-terminal domain called PR1. This is a widely distributed and evolutionary conserved family, as judged by the fact that they are members not only in mammals, but in many other individuals, indicating these proteins must play important functional roles. In mammals, 4 proteins have been described, mostly in the male reproductive tract, and 2 of them of epididymal origin, and both highly abundant within the epididymal lumen. So CRIS1 was the first identified member of this family and was described by the group of Tameo Blaquer in Argentina. It's a glycoprotein that associates with the sperm surface, both the head and the tail, during epididymal maturation, and which participates in these 2 stages of fertilization: sperm zona pellucida binding and gamete fusion through its binding to complementary sites localized in the zona and in the plasma membrane of the egg. And interestingly, there is a human CRIS protein also synthesized in the epididymis and with the same functional roles that the rodent counterpart, binding through complementary sites in the zona and the plasma membrane of the human egg. Now, structural functional studies using the entire recombinant CRIS1 protein from rodents, as well as recombinant fragments and synthetic peptides, reveal that the ability of CRIS1 to bind to the plasma membrane of the egg and participate in fusion resides in a very small region of the molecule of only 12 amino acids located in the PR1 domain, and which corresponds to one of the 2 signature motifs of the whole CRIS family. This represents the 2 domains of the protein. And here an image showing the location of the signature 2 and its accessibility. Now, in addition to the role of epididymal CRIS1 in gamete fusion through ligand receptor interactions, recent results obtained in our lab using single, double, and multiple knockouts for CRIS proteins reveal that CRIS4 plays a key role in gamete fusion, becoming another good target for contraception. And interestingly, in this regard, human CRIS1 is considered the equivalent to the combination of CRIS1 and CRIS4 in rodents, indicating the potential clinical implications of our studies in rodents. So besides the role of CRIS1 and CRIS4 in gamete fusion, results obtained in our lab several years ago in 2015 revealed that CRIS1 acts also as a novel CatSper regulator. As Mariano showed before, CatSper is the main mammalian sperm calcium channel located in the tail, composed of several subunits, and essential for the development of hyperactivation and for male fertility, as judged by the fact that CatSper knockout sperm cannot hyperactivate, and that is the reason why the animals are infertile. So electrophysiological studies using patch clam in collaboration with Alberto Darson show that CRIS1 has the ability to inhibit CatSper. And although there are several activators of the channel, this would become the first inhibitor of CatSper. So based on this, the rationale of and goals of our lab is that the identification of compounds that interfere with the roles of CRIS proteins in gamete fusion and or CatSper activity will affect fertilization and thus fertility, representing an effective, safe, and attractive male contraceptive approach. So regarding the role of CRIS1 in gamete fusion, we are looking for compounds that could interact with the signature 2 in CRIS1, blocking the interaction of the sperm CRIS1 with the egg and inhibiting gamete fusion. While in the role of CRIS1 as a physiological CatSper inhibitor, we propose to identify a compound that mimic or strengthen the binding of CRIS1 to CatSperm by interacting with the same binding site, but with a higher affinity and leading to an irreversible inhibition of hyperactivation, and that therefore to a male contraceptive approach. But of course, a compound like this could also inhibit CatSper in the female, leading to a female contraceptive. So what we are doing right now is we are working on the identification of compounds that interfere with these functional roles of CRIS1 with the support of MCI. And what we are doing basically is to develop a high throughput screening assay called AlphaScreen that senses the interaction between two labeled molecules. It's a proximity assay. In our case, the two molecules are recombinant CRIS1 carrying the signature 2 and an antibody against the signature 2. So these two molecules are coupled to donor and acceptor beads. And if there is an interaction between them, there will be a signal that can be detected by a plate reader. And this can be used for a screening of a compound library with the idea that those compounds that bind to the signature 2 will interfere with the signal, becoming potential candidates to block gamete fusion and fertility. And we proposed the same assay for the measuring or identifying compounds that inhibit the CatSper inhibitor activity. And what we are proposing is, instead of using the anti-signature 2 antibody, to use a CatSper subunit that binds to CRIS1. So we believe, in conclusion, that these studies will provide interesting preclinical information that will contribute to the identification of new and safe targets for non-hormonal male contraception. And I'd like to finish by thanking all the people in the lab present and in the past that worked in this project, our national and international collaborators, and of course the organizations and agencies that support our work. Thanks for your attention.
Thank you.